Welcome to this month's edition of the ESTIV and ASCCT webinar. These webinars are hosted by the American Society for Cellular and Computational Toxicology and the European Society for Toxicology in Vitro. I'm Christy Sullivan. I am the Secretary of ASCCT, and I'm excited to welcome you to an almost summer webinar, but we're very, very excited to have the two wonderful speakers with a exciting and maybe not often discussed topic. So just a couple of logistics and pieces of information. The webinar is recorded, and the recording will be posted on the ASCCT website a few days from now. You can also visit the webinar archive on the ASCCT website to watch the past webinars and explore archived materials. In the webinar platform, you can ask questions using the Q&A feature at the toolbar at the bottom of your screen. So just click on that, enter your question. You can even enter them as the speakers are talking, and there will be a joint Q&A after the both presentations. If you like, if you want to direct your question to one specific speaker, please go ahead and do so, or we will kind of direct them to both as they're worded. And if you see questions you really like, you can also upvote them using the thumbs up button. And I see people are saying hello in the chat, so please continue to do that. Welcome to everyone. And if I could have the next slide, I just have a couple of brief notes for you about upcoming events. First of all, the annual ASCCT meeting is coming up in October. The abstract deadline is July 14th. So feel free to get your abstracts in for oral or poster presentations. We also have continuing education courses this year, which have just been announced. So if you just take a look at ascctalks.org/annual meeting, you'll get all that information. We hope you will join us. Okay, and then I also want to let you know that you should save the date for the next ESTIV Congress, which will take place in Prague from the 27th through the 30th of May in 2024. So it's very exciting, and hopefully we will see you all there. So on to today's program. I am very excited to introduce our two speakers today. Speaking first is Schoeters from University of Antwerp, Department of Biomedical Sciences and Toxicological Center. It looks like we spelled your name wrong. Sorry about that, Greet. But Dr. Schoeters is an emeritus with assignment at the University of Antwerp, as well as vice president of the Belgian Superior Health Council. She's managed projects in environmental and health and toxicology from the Flemish Institute for Technological Research for more than 30 years. She coordinated with the Flemish Human Biomonitoring Project for many years and has helped both EU-wide biomonitoring activities through involvement in several EU projects. She coordinated HBM for EU from 2017 to 2022, which was a joint effort of 30 countries. The European Environment Agency and the European Commission co-founded under Horizon 2020. Crete served as vice chair of the Science Advisory Committee of the European Environment Agency from 2012 to 2020 and participated in a number of risk assessment committees. Of special interest to her are the use and implementation of molecular biomarkers in cohort studies, and also in vitro mechanistic toxicology to better understand the effects of chemicals in humans and improve the dialogue with decision makers and stakeholders to promote the uptake and use of scientific results. Finally, she also served as president of the European Society for Toxicology in Vitro from 2008 to 2012. Our other presenter, Mirjam Luijten, is a senior researcher toxicologist at the National Institute for Public Health and the Environment, or RIVM, in the Netherlands. Her research is aimed at next generation risk assessment. In other words, the development of better, more human relevant and more cost-effective systems for risk assessment of chemicals. Her areas of expertise include genetic toxicology and carcinogenesis, chemical mixtures, adverse outcome pathways, new approach methodologies, and human relevance. She's been involved as a work package leader in various international projects, such as Risk Hunter and PARC and HBM for EU. In addition, she's a member of various international committees, including OECD Expert Group on integrated approaches to testing and assessment of non-genotoxic carcinogens. So as you can see, we have two very highly qualified, very exciting speakers today. And with that, we will turn the floor over to Dr. Schoeters for the first talk.
Thank you, Christy, for this nice introduction. And I would also like to say good morning to the American colleagues and to of course, good afternoon to my European colleagues. It's very nice and warm here at least in Belgium. And I'm really, together with Mirjam, we are really excited to be here this afternoon and present to you the results of the HBM for EU, Human Biomonitoring for Europe project. Can you see my screen? Is this okay?
Yes, looks great.
Okay, good. Then we will start. So Human Biomonitoring for Europe, the project has been created to support the chemicals policies in Europe. And this is quite a challenge. I should move, but it doesn't work like that apparently. Oh, yes. Yeah, so it's quite a challenge. If we see that the capacity for chemical production is really steeply increasing over the last years. Between 2000 and 2015, there is almost a double. So we have lots of chemicals in our environment and to which we are exposed. Also, it has been estimated that about 10% of this big volume of chemicals which are around are either carcinogenic, mutagenic, or toxic for reproduction. So there is a big challenge ahead of us. And in Europe, there is a new policy as part of the Green Deal to go for a zero pollution and also to have a strategy of chemicals which is sustainable. The aim is to, by 2050, to reduce the levels of pollution so that there are no further concern for health or environment. This means that many of the chemicals of concern should be banned or restricted and can only be further used for essential uses. Also, chemicals of concern should be substituted by substances, by safe alternatives. And the other challenge is to tackle the cocktail effect. So this is really a big challenge for risk assessment. And as you all know, for risk assessment, what type of information we need from chemicals? First of all, the hazard identification, of course. Then we need to know from which concentration is associated with an adverse health effect, causes an adverse health effect. But also, a very essential element is what are the concentrations of chemicals to which the human population is exposed. And this latter aspect is the one which is taken up by human biomonitoring. So it's really a very important part for the risk assessment, and it also helps people who do toxicological experiments to find out which concentrations are relevant in relation to human exposure. So human biomonitoring, this means that we take a sample from humans. This can be a urine sample or a blood sample or even a hair sample. And in this sample, we measure the concentration of the chemicals or of their metabolites. So we have their direct information of the individual from which we take the human sample. These study participants also fill out questionnaires with information on their environment, their age, their educational status, their lifestyle, so that we know something about their environment and their lifestyle. The big advantage of human biomonitoring is that you measure really the concentration of the chemical or its metabolite in the body itself. And takes in this way into account what you get into your body by inhalation, and by dermal uptake and by ingestion. So chemicals from different sources. So it really fits also in the concept of one chemical, one assessment. You get a good picture of the concentration in the individual. It may be a different age group, people with different environmental surroundings. So we have really a personal dose. HBM4U has created a network of countries working together to achieve a human biomonitoring network in Europe. So as Christy told already, 30 countries work together, together with the European Environmental Agency, to bring the information on human biomonitoring from Europe together. It was a big project. It lasted 5.5 years. It only finished last year. And it was financed by the European Commission and also by the 30 participating countries. There are more than 100 partners in the project, research partners, and it was coordinated by the Umweltbundesamt from Germany and co-coordinated by us from Vito. The challenges of the project were to really try to find answers to the questions policymakers have, so to really support the chemical policies in Europe, but also to see how the human biomonitoring data can be in an effective way be used to in the risk assessment. Of course, in Europe, we wanted to also be able to harmonize our procedures, to harmonize our data, and to come up with comparable data. So quality assurance and harmonization was an important aspect, but also we wanted to have some more information and do some extra research on novel techniques to identify a wide range of chemicals and also to find more insight in the link between exposure and health effects. So this was quite an ambitious challenge. What we did first was to develop the dialogue with the policymakers. We wanted to capture their priorities and their policy questions, and the dialogue continued throughout the project in a way that they were aware what we were doing and that they were also ready to take up their results in their policies. So it was very important to clearly communicate the science to the policymakers. And the policymakers were involved both at the national level from these 30 countries and at the EU level. We learned also as scientists during the project that it's very important to provide the results when the policymakers need them. But in Europe, we were lucky because at that time, they were developing their new strategies for sustainable chemical strategy, the zero pollution ambition, the farm to fork transition. So they really were ready to take up the results. So it's very important if you talk to policymakers that you deliver the results at the moment they need it. So in the different countries, we had national hubs, and they, and together with the EU policymakers, they came up with a list of priority compounds on which we should focus in this big project. And you see them here listed. About 19 chemical groups were prioritized. And the questions which we had to tackle was, what is the current exposure in the European population? Are there differences between the countries in exposure levels, depending on where you live in Europe? Are the exposures which we measured by the biomonitoring, are they of health concern? And the policies which have been implemented until now, were they effective? Or do we need more regulations, more policies? So these were the main questions with which the policymakers came up. So what we did we created a human European human biomonitoring platform in Europe. And as part of it, an important part were the national hubs. In the 30 countries, we had groups of policymakers, researchers, and stakeholders working together and meeting from time to time. They also provided us input on which human biomonitoring studies were planned and were ongoing in these different countries, and whether we could access the samples and the data. This information on the data is now available on publicly available websites. But we also created a network of laboratories throughout Europe because we wanted that if biomarkers were analyzed, that at least that the results would be comparable between the countries. And many laboratories built capacity to biomonitor the samples. We organized also a quality control, quality assurance program so that we were sure that data from one laboratory were compatible with another laboratory. In total, 152 biomarkers were measured corresponding to 9 chemical groups. So we made a big step forward in Europe to build capacity to exchange knowledge and to make sure that we get comparable data. Another aspect I would highlight is that we also collected many new human biomonitoring data. 21 countries really worked together to align their studies and to make one big study. And the focus of this study said they could be included if their study addressed the general population, so not participants from hotspot areas and near industry, for example, or not patients group. So the general population was addressed and the samples had to be collected between 2014 and 2021, so the last 5 years. We recruited studies which addressed different age groups, children between 6 and 11 years old. Another group was teenagers between 12 and 19 years old. And another group were adults, young adults in reproductive age between 20 and 39 years old. So we wanted to make sure that the studies came from all over Europe so that we get a good picture of the exposure in Europe. And in each of these age groups, studies from the north of Europe, from the south of Europe, from the west of Europe, and from the east of Europe. Male and female participants had to be included. And we also were careful that we included people from different educational statuses, because the education people have also corresponds to some lifestyle factors. And we wanted to have a good picture of different socioeconomic classes in Europe and their exposure. And also we made sure that there were inhabitants of rural areas, of cities in each of these studies. So let's now have a quick look to the results, because I can only show you a flavor of these results. What we found for all the biomarkers we measured was that there are statistically significant differences between the study areas. So depending on where you live in Europe, your concentrations are different. And here you see an example for acrylamide biomarkers. Acrylamide is present in food, like in fries and in roasted coffee. And what we have seen is that, for example, for acrylamide biomarkers, levels are much higher in southern countries than in participants coming from other areas in Europe. If we look at the right side to perfluoroalkyl compounds, PFOS, PFOA, then we have seen that which are present in a lot of consumer products, and which are in Europe at least of a concern in many countries. We have seen that the levels, the concentrations in the population is on average higher in the northern and western countries than in the southern and the eastern countries. So this tells us that although at the European level, chemicals are regulated, there are important differences between the countries, and not in all over Europe people are equally well protected. So national follow-up is needed. An also striking factor is that we could see that for many of these short-lived chemicals which we looked at, levels where the concentrations were higher in young children compared to older age groups. And here you see an example for pesticide metabolites, chlorpyrifos, and the insecticide metabolites of pyrethroids. And you see the yellow bars are from children, the blue bars are from adults, and for each of these metabolites, you see higher levels, higher concentrations in children. This may have to do because children per kilogram body weight eat more food, and also their inhalation rate per kilogram body weight is also higher than in adults. And if they live closer to agricultural fields, they may be more exposed and have a higher uptake. But also for plastic compounds like the phthalates, which are plasticizers used a lot in polyvinyl chloride, we see again that for the different plasticizers, children have higher levels, higher concentrations than the older teenagers. This was so for all the plasticizers, and this may have to do with the fact that children play close to the ground and may ingest or inhale more dust. They use a lot of plastic toys and put them sometimes in their mouth, which also may be an exposure route. And they also have a higher skin surface area in relation to their body weight compared to adults, which also may have some effects on dermal uptake. What I want to say is that for many of the chemicals we looked at, here you see a list, we systematically could find that the younger age groups, the children, have higher levels than older age groups. This means that the recommendation from the project is that we need to have special protection for the young children. Another aspect, we have looked at changes over time. And at the right side, I will see whether I can use a laser pointer. At the right side, you see the results of a specific phthalate, which has been measured in urine of children in these aligned studies of HBM4EU. We also measured the same compound in samples which were collected about 10 years ago in the Democophis project, also with samples from different European areas. And we could see that the levels at that early time interval were much higher than now in the aligned studies. During this period, this chemical was regulated, and we could see that the levels were decreasing. This was only not the case for DIBP, but also for other phthalates, for bisphenol A, for PFOS and PFOA. All these compounds were regulated over the last 10 years. But if we look at substitutes for these plasticizers, such as DINCH, we could see that the present data from the aligned studies provided us with much higher concentrations compared to the samples which have been measured in the Democophis studies about 10 years ago. So substitutes readily enter the environment and are taken up by the population, and we see them appearing in all our study participants. Although the problem is that these new compounds are not regulated and that there is almost no toxicological information about it. So we really ask the regulators to speed up the regulation and to take into account the new toxicological information when it comes out. Another example is acrylamide in adults, in urine of adults. You see, this was regulated in 2018, and you could see that before this time period, there is a steadily increase in the concentration in the adults, while after 2018, you see the first decreases in the concentration. The policymaker is really that we need policies because it really helps to control the exposure of the population. you can consult them in public in the European Human Biomonitoring Dashboard, which is available at the website. You just select for a chemical group. Then you can select for a biomarker, for a matrix. You can specifically look at different age groups, and you get that information on for each study how the exposure in that study is. You see the different percentiles here. You can also look at the time trends. Here you see over the years the PFOS levels decreasing in the serum of the samples that were taken over time. So this is available at these websites which are mentioned here. The other aspect, the other question is, are the levels that we measure, are they of concern for health? And therefore, we compared the levels, the biomonitoring data with safe limit values. And therefore, we use the health-based guidance values. This means if your biomonitoring concentration is above these health-based guidance values, based on current knowledge, you cannot say you cannot exclude that adverse health effects will occur. If your biomarker level is below it, then at least based on the knowledge which is now available, there is no concern for health. Within HBM4EU, 27 health-based guidance values were derived, and you can see the procedure in the publication which is mentioned here. Also, all the levels are published. These health-based human biomonitoring guidance values are internal concentrations. There are also similar safe limits, for example, the biomonitoring equivalents, which are derived by Summit Toxicology and are used by Health Canada. And also in Germany, you have the HBM1 Commission. So these are expert panels who also derive these safe limit values. If we compare then the results of our aligned studies, and this is an example for PFAS, with this health-based guidance value, then you see, for example, for separate compounds, P4 and PFOS, that the teenagers of our populations, in some of them, health-based guidance values. And if you look at the sum of several of these perfluoroalkyl compounds, then even a higher share of the population exceeded these health-based guidance values. So the conclusion and the recommendation we have for the policymakers is that it's needed to consider these compounds, these PFAS compounds, and to regulate them as a group. The same can be said for the phthalates. Here you see for individual phthalates, a small share of the children in this case that exceeded these health-based guidance values as seen by the red parts. But if you look at the combination of these phthalates present in the individuals, then we have to say that about 17% of the children exceed the health-based guidance values calculated based or derived based on the hazard index approach. So again, it's necessary to regulate these compounds as a group to prevent regrettable substitution. And if we look at all the chemicals, at all the chemicals which we use in our aligned studies and for which we have these safe limit values, then we can see for almost all of these chemical groups that a part of the populations exceeds these safe limit values. So for the conclusion is that in Europe, we are not that well protected against the hazardous effects of chemicals. One thing I would like to add is that we also have looked at whether the exposure biomarkers, which we measured in the aligned studies, are in some way connected to health outcomes. And therefore, we introduced biochemical biomarkers and effect biomarkers in the same samples from the same individuals. The selection of these biomarkers was based on what we know from the literature, experimental studies, and also epidemiological studies, but also on the AOP wiki. So we looked at the adverse outcome pathways and tried to find biomarkers which we could introduce in our aligned studies. And so this is the list of potential biomarkers which were associated, which were introduced in the children and in the teenager studies in relation to flame retardants, phthalates, and perfluorinated compounds. And the basic idea was to associate it with neurodevelopment, sexual maturation, and metabolism. The studies are still ongoing, but we have some results yet. And for example, bisphenol A was associated with changes in brain-derived nerve factor and also with tau problems in our studies sample. And for the phthalates and the PFAS, we found associations with the hormone kisspeptin and also with reproductive hormones and puberty development. So this tells us that at the current exposure levels in our population, our study population in Europe, there are some signals that biological changes and adverse health effects are occurring. So the recommendations to the policymakers were that really regulation is needed, it helps, and we should be careful for specific compound groups like bisphenols and PFAS to regulate them at the group level. We also recommended that in Europe, such a sustainable human biomonitoring network would be very helpful, and we don't have it for the moment. We need and we want to continue with this, to have this network of QA, QC laboratories, to have a harmonization of our procedure, of our sampling, and of our data analysis so that we can make comparable data accessible. We also stress the fact that further research is needed. And there I want to pass the word to Mirjam, because some research has already been done in HBM4EU and will be continued in PARC. We need more capacity for looking at the broader range of chemicals. We have more data, we need more data on what is the real life exposure, what mixtures are occurring and how to deal with them. But before passing the word to Mirjam, I would like to really mention that we have put a lot of energy in our project to communicate about our results to lay people. We made fact sheets, we made infographics, we made videos. For the policymakers, we made specific policy reports. And also we have published a lot of manuscripts by now. There are also 2 special issues which combine some of the manuscripts, but you can find much more of them in PubMed, and also at our HBM4EU website, you find much more information. By this, I would like to thank you for your interest. I hope to have given you a flavor of what we have done. But I also want to thank, of course, all the collaborators of HBM4EU, but also the stakeholders, the management board, policy board, and our national hubs. And I really look forward to, of course, what Mirjam will tell you, and also to your questions. Thank you.
Thank you, Greet. That was wonderful. I have to say, your science communication and your slides are so clear and engaging. It must be because you're working with policymakers from the beginning on your projects. So compliments to you.
Thank you. Thank you.
We have some questions coming in already, but we'll move to Mirjam's presentation and then and we'll answer them all after that.
Okay, thank you, Christy. And then I also will share my screen. So, and I hope you can see it okay and hear me well.
Yeah, it looks good and you sound good. So the floor is yours.
Okay, thank you. Yeah, well, as Kate already mentioned, we also looked in HBM4EU into the cocktail effect. So that's why I chose this picture, also because of, obviously, since we're nearly heading for our summer break. So in this presentation, I will focus on the use of human biomonitoring data for the identification of real-life mixtures. And so why mixtures? Well, I hope many of you are aware that people are continuously exposed to quite a few different chemical substances from different environmental sources via different routes, and also with different durations of exposure. And consequently, we may suffer from increased health effects of these exposures because different chemicals then may add in terms of, so have an added effect when you are exposed in combination. So obviously we need to act on that and deal with this issue of mixture risk assessment. But that's way easier said than done because there is a very large number of chemicals present in the environment. And that makes mixture risk assessment really complex and poses quite a few challenges, not only for risk assessors and risk managers, but definitely also for scientists. So within the HBM4U project that Greet just very nicely introduced, what we aim to do is to really create a better understanding of the exposure to chemical mixtures in Europe, but also to have a better understanding of the resulting health effects. And all that was obviously done through human biomonitoring. And in total, we had 3 different activities, being the first one that we really aim to determine. So what are now the real life mixtures we are exposed to by making use of existing human biomonitoring data from studies across Europe. But then in addition, we also generated additional data. So in addition to the aligned studies that Greet just presented, and those data were really focused on pesticide using suspect screening analysis. And then there was a third activity that was focused on mixture health effects, for which we then also developed an advanced workflow, how to assess such health effects. But due to time constraints, I will not present on that. But nevertheless, I think you will be able to find quite a few interesting publications on that. So I will focus on the first 2 activities. And so let's now look then first into this issue of what are the real life mixtures we exposed to. So what we did in HBM4U is that we evaluated existing human biomonitoring data using correlation network analysis. And so I don't know to what extent you are or are not aware of how human biomonitoring studies are typically carried out, but normally, so there is a number of exposure biomarkers that is measured in the same individuals. And as Greet just explained, that can be in a urine sample, can be a blood sample, can even be a hair sample. But although this has been, let's say, well, the typically approach for decades, I would say, the use of correlation patterns among these biomarkers has been largely ignored. There are then different ways how you could, once you do a correlation analysis, how you can present the information. And that is, so let me also try to bring up my pointer. So here on the top, this is an example how you then can actually display or present this correlation information. But what is typically more challenging is then how to interpret these data. And so networks, of which you see an example here at the bottom, that is a really nice graphical way. That is really, let's say, a more intuitive approach to get a grip or to understand what is it now that you're looking at. So what are the results from these correlation analysis? And so that is what we set out in HBM4U. So proof of principle in terms of applying network analysis on human biomonitoring data. So that proof of principle was developed firstly using a simulated data set, and then next using a real-life data set from the Flemish Environment and Health Study. And you may wonder why we did not use real data from the very beginning. Well, that was a very lame reason. When we started with HBM4U, then shortly after, the GDPR regulation, so the privacy regulation, was implemented in Europe, which prevented us in the beginning to, let's say, carry out data analysis as we were used to do. So there we needed some time to find some workarounds to comply with the regulation and at the same time be able to do some proper data analysis. So that's why we started with the simulated data first. And that worked out actually quite nicely, although we obviously had to overcome a couple of challenges, but in the end it worked out very well. And so then we continued and applied this network analysis to data sets from 4 different HBM studies, being studies conducted in Germany, Belgium, Spain, and the Czech Republic. And each of these studies, well, were quite different if you look across them, not only in terms of what were now the exposure markers that were measured in each of these studies, but they were also quite different in terms of study design and population groups involved. So some studies were more focused on workers. For instance, the Czech Republic study was focused on firefighters. Well, for Belgium, it was a different cohort, by the way, than the Flemish Environment and Health Study. And then for Germany, it was a subset from the German environmental studies. And so obviously I cannot present all the outcomes and the findings from these studies today, but I nevertheless want to show you an example. And so this example that you see here on the right is the outcome of such a network analysis, so carried out for a data set from a JARIS-5 subsample. And so in this subsample, we use data from participants with the age of 3 to 17 years. And the data were for biomarkers measured in morning urine samples. And so this is then the network that you get out of such a network analysis. And then with the different color codes, we indicate the different, what we refer to as communities. So these are groups of correlated exposure biomarkers reflecting co-occurrence of chemicals. And if you look a bit more closely, you may be familiar with some of the abbreviations typically used for different exposure biomarkers. So for instance, here, this green upper one, so that is community number 7, that is mainly comprising PHHs. But then we have here, so number 1, these are 2 parabens, whilst this third one is then lilial, also known as lismorol, which is a fragrance. So this may be due to exposure to cosmetic products, for instance. But then we also saw, for instance, here, this community number 2. So this is acrylamide and its main metabolite, but then it apparently also correlated to a benzene metabolite, an aprotic solvent. And then here, these are the methyl isothionosolenones, very difficult name. So that's again a different type of chemical. So obviously what you learn here is, so which are now the chemicals that are in real life, because this is from a urine sample from different participants of the JARIS-5 sample. So these are co-occurring together. We also learned that apparently some of the chemicals are not part of a network or a community. For instance, here on the top, we have bisphenol A, but we also have mercury and arsenide. And so when looking then in more detail into this particular finding, but obviously also into the findings from the studies from the other countries and looking into more detailed analysis. So obviously you can then also do analysis, for instance, trying to discriminate between, let's say, the younger participants in the JRS5 study, so up to the age of 10 years, for instance, versus the somewhat older ones, or let's say trying to differentiate between participants with a higher or a lower BMI or educational status, etc., etc. So we learned quite a lot from these network analysis. But I think one of the main learnings was also that clearly combined exposures to chemical substances are very common. And these occur in all the population groups that we looked into. So I think that is a real, let's say, strong confirmation that indeed mixtures do matter. And although it's very challenging, we should tackle all the challenges associated with mixture risk assessment. Maybe not all, but at least many of them. We also learned that the communities that we identified in the studies, obviously, partly are maybe not so exciting. So you may say, well, you identified a cluster of PHHs, or there is a cluster of phthalates. Tampi. I mean, that's maybe not so surprising and exciting. But at the same time, in the different studies, we definitely also identified communities that consist of substances from not only different chemical classes, but also where we know that the chemicals are actually regulated under different regulatory frameworks. And so that poses a challenge in itself. And then we felt, well, of course, it's great that this works out so nicely, but then we should at the same time also be realistic. I mean, mixture risk assessment is already complex in itself, and it's by no means to be expected that whatever regulatory agency will look into each of these communities in much more detail. I think we simply don't have the resources for that. And so there we felt, so we should then integrate the outcomes of the network analysis together with toxicological information as well as concentration data to really allow for further interpretation of these outcomes from the network analysis. Because that would tell us then, so which communities are actually of higher concern. So where is there a potential health concern when it comes to a combined body burden? And so for that, we then applied just on one of the studies, being again the JRS5 study, we applied the hazard index approach. So in mixture risk assessment, that is a very common approach, which assumes dose addition, for a particular combination of chemicals. And so in order to be able to apply this hazard index approach to HBM data, we need HBM health-based guidance values. And so Greet already introduced these to you. So we derived quite a few of these values within HBM for you. But Miranda Low and Yvette Christophe de Vries from the Institute of Occupational Medicine from the UK, they actually put together a database with all kinds of HBM health-based guidance values as well as equivalents. And these values were then used for deriving hazard quotients for each substance for which we measured exposure biomarkers in the JRS5 subsample. And so these hazard quotients are then very simply calculated. So you have the concentration for your particular substance and you simply divide it by the HBM health-based guidance value or its equivalent. And then, as a next step, you can then derive the hazard index across substances for each of the individuals in the study, simply by summing the health quotient values for each of the substances. And so if we then go back again to this example that I just showed you, so the outcomes from the network analysis for the JERIS-5 subsample. So what I indicated now as well in this figure is the substances for which we actually could identify an HBM health-based guidance value. So these are marked in green, and sometimes the lines are dashed, and that means that actually for that particular substance, there was not a specific HBM health-based guidance value, but there was only HBM health-based guidance value derived for a particular combination of substances. And so if it's dashed, then actually the value is reflecting a combination. And then for some of them, for some of the substances, we did not have an HBM health-based guidance value, but we only had a biomonitoring or biological equivalent, and these are the ones marked in orange. And so as a first step, obviously we want to check whether indeed mixtures do matter. So we looked at the hazard indices using all the substances for which we actually had the HBM health-based guidance values or an equivalent value. And so here you see then the frequency, and here are the hazard indices. And what you can see is actually that all the participants from this JERIS-5 subsample, so that is 550 people, all of them had a hazard index above 1. And that is only taking into account the substances for which we actually had an HBM health-based guidance value or an equivalent. So in real life, if we would have these for all the substances that are included here, hazard indices would even be higher. And then as a next step, obviously what this was all about was to identify, so what are now the communities posing a potential health concern. And so what I show you is just one example. And so that is this one, community number 1. And so there we applied, again, the same exercise. And that told us that actually 5.8% of the participants, so nearly 6%, had a hazard index above 1, which is considered to be of a health concern, whilst the majority were below 1. And similarly, we did it for a couple of more communities. Obviously, if you are interested in mixtures, if we have a community with only a single health-based guidance value like we had here for the PHHs, it doesn't make sense to apply this approach. So for the other communities, it was well below 1. We also learned in this exercise for all the substances actually that here the main health concern is really driven by arsenite and by acrylamide. So that's also a very interesting learning. So obviously there is still a lot more to do. We felt there were quite some challenges still to be overcome. We were clearly also limited in our exercise here by having just health-based guidance values for a limited number of substances, but nevertheless, we felt we demonstrated the proof of principle that indeed toxicity weighting can be applied at the level of identified communities, which then in the end may also help to identify, well, which are now the communities, the combinations of chemical substances that we need to act on. So that really helps us to prioritize. And so in the final part of my presentation, I will focus on this second activity, which was about generating new data on pesticide exposure using suspect screening analysis. And so for this, we developed and implemented an additional survey. So in addition to the aligned studies that Greet just presented, and so that is a survey that we named Survey on Pesticide Mixtures in Europe, briefly referred to as Specimen. And so with this study, we aimed to generate new exposure data on a broad combination of pesticide. And we also aimed to assess whether there are possible local contributions when it comes to pesticide exposure. So, and more specifically, the research questions were as follows. So obviously, what are now the combinations in terms of pesticides that we most often detect? Can we identify hotspots? So do people that live close to a pesticide application site, so more in a rural area, do these folks have a higher exposure level compared to people living further away from a pesticide application site? If so, can we then also find out whether there is actually a difference between seasons? And here we do obviously refer to a spraying season versus a non-spraying season. And are there now then also differences between age groups or perhaps in study populations? So what about the pattern across Europe? And so to explain the design of the specimen study in a bit more detail, so we collected samples during both the non-spraying season and the spraying season in households that we refer to as hotspots, so living within 250 meters from an agricultural area, as well as from control households. So it was a urine sample that we collected, both from children as well as from one of their parents. And here the children really had the same definition as used for the N9 studies. So children were in the age of 6 to 11 years old. Besides collecting a urine sample, people were also asked to fill out a questionnaire. And initially, we also aimed to collect house dust, but unfortunately, due to the COVID-19 pandemic, there was not something we were able to complete for all of the samples in all of the countries. So that is basically the study design. And then the urine samples were subjected to a consolidated and harmonized methodological workflow for suspect screening. And there was really a major effort in HBM4U. So there was a dedicated work package for this, so dedicated to emerging chemicals. And so under the supervision of Jean-Philippe Antignac, this workflow, this harmonized methodological workflow was really established in HBM4U in different laboratories. And what I forgot to mention is that we carried out this specimen study in 5 different countries, being the Czech Republic, so indicated here in pink, Spain, it's more greenish, brown-green, Hungary, Latvia, and the Netherlands. And applying then this workflow for suspect screening, in total, 95 pesticide-related markers were identified across the urine samples, so all using the very same methodology, and nearly half of them could be identified with a high level of confidence. So if you annotate the different suspects, so to say, then there is a dedicated approach that allows you to then assess, so what is the level of confidence that indeed this substance is what you think it is. And so for nearly half of them, we were quite confident that it was or is what we thought it was. Well, given that we were measuring in a human biomonitoring sample, then obviously what you measure is both the parent compound as well as the metabolite. And so by digging deeper into these 95 pesticide-related markers, we found out that actually these relate, the ones that we could identify with a high level of confidence, so that's about half of them. So these relate then to 29 parent compounds. And these are very well-known pesticides. And what you then can see here from this graph is really, so what are now the number of what are the combinations that we observe, and so what are the numbers in terms of different pesticides that we are exposed to. So that is quite different. So as you can see here, for instance, in Latvia, there were not that many combination of that many chemicals, pesticides in a combination measured, whilst, for instance, in Spain, the number of parent pesticides measured together in a urine sample was way bigger, way larger. So, and then we also looked into the data a bit more, obviously trying to address each of the, or figure out each of the research questions I was talking about earlier. So what I only want to show now today is, so what you see here is actually a frequency plot. So what is now the frequency of co-occurrence in the urine sample? Again, using the same color codes for the different countries. And so here you have a listing of the different pesticides. And to cut a long story short, so we found out that there are quite some differences in detection rates when it comes to hotspot versus the control areas. There were also quite some differences between summer and winter, or let's say spraying versus non-spraying season, and even also when it comes to differences between children versus adults. But in many cases, those differences were not significant. And that may be very well due to the fact that this study was designed and implemented in conjunction then with the aligned studies. So let's say the study involved not that many participants per country. It was about 100, so including adults and children. And then, of course, each of the participants was sampled twice in the spraying and the non-spraying season. So altogether, that is not a that large sample size, which may also then have prevented us from finding some significant differences. Or maybe this is what it is. That is something that we hopefully will also learn more about in the PARC project. But nevertheless, if we then look at what this study told us, so we felt it's really a valuable approach in terms of learning more about what is the combination of chemicals that a given population is exposed to, and that really helps us then also to, for instance, use such an approach to get a feeling for, so what are now substances for which we actually should carry out a targeted approach. So by using the more, let's say, traditional approach in terms of measuring a specific effect biomarker. Typically now that is really, well, the chemicals that we are very well aware of, and I think for a good reason, but obviously with changing chemicals spectrums being used for different types of products, it's really good also to have a better understanding of, so what are now indeed then the chemicals, so the emerging chemicals of concern. And for this, suspect screening is really a valuable approach. Well, as I already mentioned, in this study, so in this survey, we did not observe strong contributions from agricultural use of pesticides. But again, that may be hampered due to the fact that it was not such a large study. Overall, and I think this last conclusion actually applies not only to the suspect screening, but to all the work we did, is so mixture risk assessment could really benefit from having, let's say, a more harmonized strategy when it comes to measuring both exposure and effect biomarkers in HBM programs. So as Schoeters told you, we have already quite a good program, HBM program implemented in Europe, but until HBM4U, that was not as harmonized as it should be. And hopefully this will be, well, let's say further advanced with the implementation of PARC. Finally, I would like to share with you that, well, looking then at all the activities we carried out in HBM4U when it comes to chemical mixtures, so for the different topics that I already mentioned earlier. So we had a good look again at, so what are the different outcomes for each of these activities? And we used those to not only draft conclusions, which obviously are also reported in each of the deliverables and publications, but we also used these to draft recommendations. And then following a series of webinars where we explained in much more detail, let's say the technological aspects of the different activities carried out, we then discussed in a stakeholder workshop these draft recommendations, and we tried to, let's say, refine them. Some of them were dropped, a few, but it was really helpful in order to get a grip on, okay, where is it where we stand now, and where should we really invest further on to advance the area of mixture risk assessment. And that resulted in a total of 14 recommendations for mixture risk assessment. And I'm really pleased that I think nearly all of them, if not all of them, are actually taken up in PARC, in amongst others, work package 6 and 4. So that is really good to see that work continued. And so there are a couple of useful links that you will be able to access later on. So once the presentation and the recording is shared from this webinar, so that hopefully that is helpful to you as well. And with that, I would like to thank you for your attention, for making time on this Friday afternoon or Friday morning. I definitely also would like to thank all the partners that contributed to the mixture activities in HBM4U. It was a real pleasure, sometimes challenging, but always pleasant. And I would also like to thank all the participants from the HBM studies that allowed us to do all this research. Thank you.
Well, thank you, Mirjam. And thanks for that additional presentation. I feel like we got a really nice overview of several different activities and different types of studies happening related to biomonitoring. And I came up with a lot of questions, but now we have a lot of questions from our participants, so we will see if we can get through them all. So let me see here. We have, first of all, we have a question to greet. Sort of an overall question. This seems to be a very successful project, which had to have a harmonized collaboration between people with different backgrounds. And cross-disciplinary work is very challenging. Can you share maybe some keys for success?
This is a very interesting question, and it's good that you appreciate it. It's not so easy to work with so many partners together. And at the beginning, it took some time before we really find out how to do it. But in my view, the key to success was that we had this continuous dialogue with the policymakers, which really directed the research towards what they needed for better protection, for better regulation of the population. And that's what all the researchers were interested in. And we learned that working together was helpful to bring these results to an action of the policymakers. And I think that's, in my view, that was really essential to have this continued dialogue and knowing that we are aiming towards the same goals, which are relevant for protection of health of the population. I don't know, Mirjam, whether you think it's different or...
No. I fully agree to that. Yeah, it was indeed in the beginning. I think sometimes also, as you often see, it's people speaking different languages. Yeah, but it was. I think the most important was that it's really beneficial to have then, let's say, different communities or different worlds working together. That's really. I think that's something we are kind of obliged to.
We learned to see the advantages of the collaboration, and that was really obvious, I think, for most of us.
Yeah.
Great, thank you. Another question is related. I think it could be directed to both of you, actually, but we'll start with Mirjam. Are the health-based guidance values used derived from oral exposure or other exposure routes, for example, inhalation?
Oh, that's a good one. I think they are sometimes also derived for other exposure routes, but then it's really also dependent on, let's say, what is the intended or the main used route of exposure. But I would guess that the majority is probably on a oral basis, and I wouldn't know whether there are sometimes, let's say, route dependent health-based guidance values. I don't think we had that luxury. We sometimes had the situation where there were different values for one substance, and then we really used the one that was most recent. I think that was the main driver. And if they both were recent, then I think we said, well, let's focus on Europe.
The strategy to derive them is published. So I mentioned publication. And if we have the choice, we go first for epidemiological data, where you have the biomonitoring data related to health effects. For cadmium, you can do that. Those were already, so this was independent from the exposure pathway. But for the rest, Mirjam, we took indeed the toxicological reference doses, if they were recent, or the tolerated daily intake doses, which are also most of the time not dependent on the exposure pathways. In some, if we don't have recent ones, then we went to the toxicological, the experimental data, and then we had to choose the most relevant exposure route, of course. So, but we have a really tiered strategy in how to derive those HBM guidance values, which can be consulted.
Yeah, and I think it's fair to say that I mean, it's not drastically different from approaches used by other agencies or other groups, for deriving these.
Yeah, and I know that they don't, as you showed in your work, they don't always exist for some chemicals.
That's true, and they change over time also. If more information becomes available, we see them often changing. So it's not absolute.
No, but I think still I was quite surprised to learn, for instance, that for arsenic there is none. Felt like, well, we know quite a lot.
Yeah.
But, well, at least we couldn't identify it. Let's put it like that. Disclaimer. Yeah.
So there's a question about hotspots, and Mirjam, you answered this a little bit, but so maybe we'll start with Greet. What was your criteria for defining a hotspot? Is it the same as what Mirjam presented, or are different studies looking at hotspots differently?
In our aligned studies, we wanted to have a picture of the general population in Europe, and to bring studies together that did not focus on hotspots. Because then you have high values, but you know the source. But the question was the overall population in Europe, how is it exposed? And therefore we avoided people living in a specific hotspot. So to include the studies, the participants should be randomly recruited and not from a specific area. But indeed, for the pesticide study, it was different. We also have studies in occupational settings, they were also more hotspot related. So but for the general population, we choose specifically not to work with patients or not to work with people living in real hotspots. So, yeah.
Do you have any idea for, looking at a population that is excluded because or I guess what percentage of the European population was excluded because they were living near a hotspot? Do you have?
I don't think you have to see it like that, because we did not exclude a population, but we collected studies from, for example, from Germany, and the requirement there was that the people were randomly included from Germany. So some may have lived next to an industry, but we have no oversampling next to the industry.
Okay.
So it makes a difference. So we wanted to have a general picture of Europe. So some people may live next to agricultural areas, others the people live in Europe, but we did not oversample in specific locations. That was in the pesticide study, in the specimen study, you oversampled in near agricultural fields. So it was different.
Yeah, and there we knew for the controls that they were actually not living. So that was really controlled. People actually live close or do not live close to the fields. Yeah.
Okay, that makes sense. So there's a question that I also, and both of you maybe alluded to a little bit, wondering about follow-up. So of course, these studies are snapshots. Is there a coordinated plan for continuing to follow up and do additional biomonitoring regularly? And maybe that's part of the PARC activities. So maybe, Mirjam, you can say something about that, but maybe we could start with Griet. What's kind of on the horizon for more studies?
I think, for the moment, the studies which were involved in the aligned studies, for example, some of them were part of longitudinal cohorts, but there is up to now, not a general strategy to make a longitudinal study in Europe for the moment. Nevertheless, in PARC, or certainly we have an initiative which is called RIVM, which is on the horizon, I would say. And there we may go for longitudinal cohorts also over Europe. But for the moment, we will be already very happy if we can at regular intervals do these cross-sectional studies and get information of the participants on their environment, on their diet, and on their health status. That's the way how we try to deal with it now. So No, please, Mirjam.
Yeah, and I think then in addition, indeed, we also continue then in PARC. Mm-hmm. So also again with aligned studies, and I think a main driver really has been HBM4U and the findings, but also as Greet showed, the value of getting a grip of if you have a measurement from a longer time ago and then fairly recent, and then maybe in a few years from now, you can really see what's happening rather than indeed only having this snapshot. And I think then making sure indeed that we all use the same method for given exposure biomarker, then really also allows you to compare, because let's say in the older days, then if you had two values from different places, whether it's in Europe or in the United States or wherever in the world, and you are not exactly sure that the same methodology is applied, then you wouldn't know with that much confidence that if you see a difference, whether is that a technological difference or is it actually a difference in exposure levels. And yeah, I think one of the additional learnings then from HBM4U is also that we now try to measure a larger number of chemicals in all of the participating countries, because now you could express interest, as in I'm interested to contribute to the aligned studies, for chemical A, B, and C, but not for X, Y, and Z. And then, of course, you still can allow, or you can perform these comparisons across regions. But it's still then, yeah, it may also be influenced by the specific countries, and hopefully this will now also be overcome with the PARC initiative. And in addition, so the suspect screening is definitely also again involved in the PARC. So there's a whole work package dedicated to exposure, with a lot of human biomonitoring, both in the traditional fashion, but also using a more innovative approach.
Right. And maybe if I could follow that up, because someone else is wondering, when you were presenting the community groups of chemicals, is that meant to kind of prioritize mixtures for benchtop work or assessment or analysis?
Yeah, I think we could definitely use it also for that. So I would say, firstly, this really helps us to identify, so what are now actually combinations that you find in a real person, rather saying, well, you may be exposed to this combination, but perhaps you're not. So really to get a better feel for what are the mixtures that we are exposed to. And then I think, yeah, it would really help to also look into, let's say, in more detail than if you look into the different components of a mixture, then obviously what should also be taken into account, but what we didn't manage simply because of time constraints in the HBM4U project, is to take into account mode of action. So which ones do have then a similar mode of action? I don't think we always know what the mode of action is. Chemicals may also have different modes of action. So there I can imagine also that it would help then to say, well, if you know what are the communities that seem to be, well, posing the highest concern, to look into then also in benchtop work, as in, so what might be an effect. And there I think, and I also saw another question, and obviously one should also, when possible, link this to epidemiological findings.
Yeah.
So what are then the health effects? Because theoretically, well, even if you then already, let's say, select a couple of chemicals and then maybe prioritize a particular mode of action or just a selected set, still there is probably too much we can do or should do. So there I would definitely also recommend to link to epidemiological findings and make sure that we then make wise choices.
Yeah. A little bit, you're sort of answering another question, which was related to kind of the benefit of a hazard index over all chemicals, or whether kind of grouping by molecular target. And I think you heard you say, like, we do what we can, right? Where you know the molecular target or the mechanism, that makes sense to do, but not always possible.
No, indeed. And I fully agree with the comment that doing a calculation of a hazard index across all the substances is indeed not really helpful. But it was more for us, as in, if you then have all these data, then it was, well, one could say, I think, scientifically a fun exercise. The outcome, I think, is far less fun because indeed, well, then we may actually risk from health effects due to the exposure to mixtures. And I think that is a serious issue.
Yeah. So a question for both of you about metabolites. And so I'm wondering, maybe we'll start with you, Greet. Looking at pesticides, maybe there are different pesticides that turn into the same metabolite or same set of metabolites. Do you have any wisdom, or did you try to take that into account in your analysis?
Yes, you're right. As some of the metabolites, and certainly for the pyrethroids, which we were looking at turn into the same metabolites. So we have to be careful for the interpretation. That's completely true. And sometimes we also have to sum up the metabolites. For example, some of the phthalates, they go into different metabolites, but they are from the same parent compounds. So it's quite complicated to take that in account, but as far as it was possible, we have tried to do that. But indeed, it would be nice to have specific metabolites for each of the parent compounds, which is not always the case. Sometimes you have endogenous substances which give rise to the same metabolite, and then you have to be careful for the interpretation. But that's reality. So we need more specific metabolites, that's for sure. Mm-hmm.
Yeah. Mirjam, anything to add?
Yeah, I agree with Greet. So we would, let's say, from a human biomonitoring perspective, we would need them. I don't think that is something that manufacturers take into account. Yeah, when it comes to also the specimen study, there was definitely that we are aware of that. It's a well-known phenomenon, also for pesticides. And that's also why, let's say, the parent compounds that we identified with high confidence, so I think there were about 40, and indeed these related then to a smaller number of metabolites, simply indeed because some of them are they have the same metabolites, so that is something you need to be aware of. And indeed also when it comes to interpreting, what is the source then? Because they also see that pesticides are sometimes used for very different crops, so that is also something to take into account.
Yeah. So maybe I can use the chair's prerogative and we'll ask one last question, and we'll start with you, Greet, because I really enjoyed the sort of a couple of your later slides where you were talking about the biomarkers of exposure, using the AOP wiki and kind of molecular information to link to biomarkers of effect. And I'm wondering if if you had a wish list for the AOP community about what kinds of things they should be doing to be more helpful in that kind of exercise, what would you say? What should be in the wiki that you didn't find or that you'd like to see more work done on?
I think it's very valuable, and I expect that it will be done in PARC. That is, Toxicological Society comes much closer with the biomonitoring community and with the epidemiology. So I think this is really the way we have to go forward. And it was a kind of a trial to see whether the wiki, the AOPs present in the AOP wiki could be useful, and they were, but you see also that there are many gaps in the knowledge, and it was only very few which had enough significance to be really taken on board.
Yeah.
So I think it's really needed that toxicological people put the information they have into these lines and develop so that the information comes together. I think it's a wonderful initiative, but there still needs to be a lot built there. And we have worked on the phthalates, for example, and it worked quite well. So we came up with some new aspects which we could then introduce, like the kispeptin, which we introduced in our course. It seems to work, so it's really promising, but there is a lot of information still lacking, that's for sure. Yeah,
agreed. Well, Mirjam, I know you agree with that. Being heavily involved in the AOP program. It is 11:30, so I want to let you all go. I first want to express my sincere thanks to Greet and Mirjam for your presentations, for sharing your work with us. So close to summer. Thank you for giving the presentation, and all of you listening for all your great questions and discussion. As we said, the webinar recording will be up on the website shortly, as well as the slides, and both of them have done a really nice job of putting some links to presentations, or sorry, links to publications and references to publications in their slides. But please feel free to get in touch if you have any questions. And thanks again, and enjoy your summer, everybody.
Thank you.
Thank you so much.